Good. Thanks for everyone coming. I know lunch was probably more appealing to be at, possibly. My name is Bryan Caron. I'm the Director of Operations and Development based at McGill University for the CBRAIN Distributed Computing Platform. I'm also co-PI of something that I'll make mention. McGill initiative, you might have heard in the opening remarks this morning about Healthy Brains, Healthy Lives. One of the core infrastructures that was funded out of that funding envelope was also something called NeuroHub, which is more of a collection of ecosystem of tools that are brought together, and CBRAIN is one of the main pillars of that platform. But today we're just going to maybe go through just some general background about what is CBRAIN, but then we'll do a portion that's going to be hands-on. You're welcome to participate in that piece as well. You don't need to have an account already. I already have generic accounts that people will be able to use to do some of the basic exercises. But if you've done any of the CBRAIN tutorials in the past on prior HIBALL BigBrain workshops, this might seem familiar to you. But with try and show in some, I would say, further developments that have been ongoing in CBRAIN that, particularly in the light of some of the things you might have heard about today in terms of single cell transcriptomics and so on, then you can maybe see some adaptability going forward in how you do things. So we're going to start this, like I said, just a general introduction. So maybe if you've been to the other ones more, there's more back and forth. This one, I'll just start with some foundation pieces and then we'll get into the exercises. And that's the first piece is just more the introduction. And typically what people have choices of doing, right? When you're working with your data, particularly since you're at the BigBrain Workshop, working with BigBrain data, you're going to be working with it. And you usually want to be able to find a way that, okay, you're not just working with one particular slice, you're working with an ensemble of data, you're bringing in other data that you're trying to integrate with the BigBrain analysis. You want to be able to have it scale, right? You want to be able to also make it such that the work you're doing is reproducible. And then you're obviously going to be wanting to collaborate and share with your peers and your colleagues with this. So of course, there's choices historically, and even still, depending on the nature of what you're doing, you might be working a lot on the command line, you might be working in notebooks, depending on how you're progressing things. But what we try and also do with CBRAIN is particularly provide a web-based interface that allows people to more focus, I would say, on the scientific endeavor rather than maybe some of the more technical computing components. That being said, there are still ways you can actually work with those technically oriented computing components from the CBRAIN point of view, which I'll touch on afterwards. But the web UI approach is what we're going to go through some of the exercises today, and to try and show you some of the benefits and utilities of CBRAIN to address the markers on the bottom, really, right? Which is it lets you have easy access to the data. It helps give you methods by which you can actually have then your work being reproducible. It can scale very easily, and then you can also collaborate and share. So we'll touch on some of those elements today. So it really is that transition from going from the terminal-based and the shell-based access, which many high-performance computing systems typically are the minimum that you're going to get, and even the maximum what you're going to get, depending on how they're configured and the policies of those particular resources. So we then provide now through CBRAIN that method by which you can transition from that terminal to web UI. And to do those things, it also not only applies to brain imaging data. The way CBRAIN is built is such that it's agnostic really to the scientific endeavor. So we've also applied it, and we're working with some colleagues in the University of Ottawa to apply this not only from brain, but also to the heart. So there's a brain-heart interconnectome project that's ongoing. So this just shows on the left some, I would say, the imported cardiac imaging data that's been brought into CBRAIN for analysis, and there's tools being constructed for that integration. It also applies to more on the transcriptomic side. I'll touch on some of the single cell transcriptomics tools that have already been brought into CBRAIN. Again, given that it's fundamentally agnostic to the actual science. And we also do other ones more oriented toward EEG, for example. That's another one that we have. So there's a wide variety of modalities and different data types that can be worked on within the CBRAIN context that we'll touch upon in a little bit today. So really what that workflow that you're going to get within CBRAIN is you're coming in through the web UI, as I said. You're going to be able to work with your data in what's called a data space, which is organized at the project level, which you'll go through an exercise and you'll create those projects yourself. You're going to then be able to select from an inventory of tools. All these tools are containerized using something called Apptainer, which you might have heard in other names, Singularity. Is anybody using containers already in any of their workloads?
Dockerized?
Yeah, Dockerized. Docker is usually, I would say, a different step, but you can then take your Docker container, your Docker image, then put that into Apptainer or Singularity image, which are typically more suited for running on high performance computing systems. Or Docker is usually the foundational step to do that. Anybody else using containers of any kind? Are you mostly using notebooks?
Yeah.
Notebooks for your case? Anybody else? Okay. We'll go through, like I said, and we'll cover some of those and maybe give me some feedback in terms of what tools you're using. But from the tools, you'll see an inventory of tools and it's arranged by categories. People can then select the tool for the data they want to analyze. You then make a decision, okay, possibly you don't even have to decide where you want that tool to run on your data. And then it can be something that you brought up to the system yourself into that data space, but it also can be brought from any of the connected data repositories that we have. Right, so there's at least because it's foundationally in Canada, it doesn't exclusively need that. Things from the Canadian Neuroscience platform. We also, as was talked about, access to UK Biobank for McGill-based members. But because we're using something called Data Lab as a technology in the back end, I have a later slide where also anything, if you're working with open neuro data sets, anything within open neuro can be accessed to the tools within CBRAIN. And very soon we're going to have a connection into a prototype we've got so far works for any of the data sets that are within the eBrains context as well. So there's a plugin that adapts it from the eBrains data knowledge space into Data Lab, which then we can import into CBRAIN. So we've got a proof of concept working for that. So this is the kind of typical workflow that you'll go through. The part on the bottom right is really, I'll touch upon it afterwards, is that if you're working with notebooks, it's not an all or nothing scenario, meaning you have to use the web UI. If you're doing things within a notebook, you can still work with data that's in CBRAIN because we have an API layer that lets you see and register and communicate with CBRAIN to move data in and out. You can even use through your API calls within your notebook, you can actually say, well, I know this tool is in CBRAIN. I can then launch that tool from within my notebook, watch its state as it goes through the entire system, and then work with the data when it's created on the back end. So we have some larger, I would say, larger projects that are typically more what we call power users, who then use a lot of the notebook environment, but they interact with CBRAIN to manage their end-to-end workflow. We won't cover some of that today, that's a little bit more advanced, but we have some examples online on our GitHub pages that give some details of how to go through that exercise. Okay. The nice thing about this as well, I won't spend too much time on it, just to say that there's other obviously elements within our ecosystem, one being multimodal data capture, collection, and management. It also comes from our lab at McGill and Alan's space, where we have multimodal capture, as I said, for imaging, also for the genetics and epigenetics, the physiological, even managing biospecimens. That can tie into also CBRAIN as well. And we have some projects that do that full end-to-end integration for the tools on the CBRAIN side. So it's been very, very scalable and very capable. What we'll see from some of the exercises that CBRAIN will provide through that web UI. This might be too small on your screen for people here, but if you're outside, you'll see that it provides a single kind of pane of glass view, as we call it, that lets you see the aggregation of all your storage that you might have connected in there. CBRAIN doesn't centralize the data in one place. CBRAIN is a federated data space and a federated compute space. So it can be connected to your lab-based servers. It can be connected to high-performance computing systems. It can be connected to cloud-based resources that your institution may be using. So all that is connected to CBRAIN and then made visible to you through that single pane of glass view that I described. So you can see it all in one way, even though it might be connected to multiple varied systems. So that's not only for your files and your data that's there, but also for your compute. And the nice thing that from the CBRAIN point of view is that if you have any, say, visualization requirements, we have a built-in visualization tool for surface and volumetric viewing called Brain Browser that was started within CBRAIN a number of years ago. That is directly embedded, so that way there's no download necessary to your local laptop, your workstation, to actually do the visualization. You can just look at it directly within your web browser. You don't have to download and move the data that way. And of course, once you have the data, you've visualized some of it, you make some decisions about what you want to run on it to analyze it. And within that same view, within the web browser, you're able to select the files, whether it's one or even thousands, to process within CBRAIN and from a menu of tools that we'll go through the exercises here. This is how, example, it was done internally for our McGill-based UK Biobank project. We pre-processed all the UK Biobank data using things, tools like FMR Prep, for example, and then we shared that output to all the project collaborators internally so that they themselves didn't have to run that computational exercise. So we just managed the workflow ourselves using CBRAIN. So just to give an overview of the impression, yes?
So you were saying that you have some HPC and cloud power. Is it bound to the CBRAIN, or do we have to have our own?
We have some general compute resource that is available to the entire community. That's part of our resource allocation on the HPC systems in Canada.
Okay.
So that's accessible to everybody.
Okay.
That, in addition, as I'll talk about in a couple of slides, we also connect to other HPC resources, including ULICH on the Joule system, for example. So if you have resources at your institution or on a system, we can work together to actually have that interface to CBRAIN so that you can then leverage it within the CBRAIN platform. It doesn't mean it's open to everybody else. It's explicitly restricted for access to you and your team based on the rules. So if it's an allocation for a specific resource, it would be mapped to accounts that would only be accessible as you deem appropriate.
Okay, and then, for example, in order to have access to the Canadian investor to come to the Canadian group, or
no. We have, for our particular, obviously, if you're doing some specific collaboration things with the group at McGill, then you can have, let's say, more enhanced access. But we also have a certain portion of the resource. We make it available as a general community resource. So, for example, all researchers in neuroscience who can access that allocation.
Okay.
Of course, we don't give everyone the maximum available because we have to serve this as to the community. But we see wide variations of users. We have users from all over accessing that common allocation for their individual work. But then we also then moderate the access to have, I would say, more specific strategic collaborations. But yes, you can access that Canadian-based one, in addition to bringing your own resource as well.
Yeah. Okay.
So just to give you an idea of some of the tools that are there, there's a long list. If you go to the URL that's posted on the screen there, we have it automated. You don't have to have an account to see that full list. It's not organized as nicely as this, but what you can see is two different menu options. One that'll show you the tools listed alphabetically, but also the type of tool that it is, organized by what we call tags. It's just a way of describing what type of modality you might be working with and what type of tool it is. Then we also have a separate tab about data sets to show what data sets are immediately available there as well. But the fundamental thing is you can see there's a broad spectrum of different tools that are available, but it's all dependent on the key elements of that. We containerize the tools. If we containerize ourselves, it may also be a container that's been given to us directly from the source creator. So if it's the source developer author that provides a container, we try and as much as possible stick with that container. We use something called boutiques to really just a JSON file to help map the inputs that you would normally type on the command line, but to the web format. So it's really a translator that presents then in the web UI what all the parameters are, and you'll see that through some of the exercises. And then CBRAIN, we just orchestrate the workflow of taking the input, running the tool, and producing the output, and then have that linkage in between. And I'll show you some of that. So you can see some of the main categories here. You may, again, take a look at the link and make it see more specific. These are just some of, say, the main tools that are within each category. And like I said, the ones that we're particularly adding, we're adding more tools all the time. But the particular ones, maybe just to emphasize as well, as on the genetic side, the single cell was mentioned in some of the earlier talks today. We've added some single cell transcriptomics tools also to the mix because of particular research activities going on that need some of these tools. So those are being used as well. One of those, for example, is cell ranger count. But the nice thing is, again, it exercises this end-to-end workflow, right? People are using the GUI, picking the data, selecting where it wants to be run. It takes the input FASTQ files, but also the nice thing is with the embedded viewer that we have within CBRAIN, because it's just producing an HTML output, cell ranger, or if you have a tool that produces just HTML output in general, You can view it directly within the CBRAIN web interface. You don't have to download it separately to look at it directly on your computer. You just can embed it, see the embedding directly within your account on CBRAIN. So for example, this view of the output from Cell Ranger is directly visible within the CBRAIN portal that we use.
Would you download it if you wanted to?
Yes, oh, absolutely. You can download either the entire output, you can download subsets of the output. You have that flexibility there to make a decision about what you would like to just, it's not all kept there and locked away. You can do with it as you want, but it just provides that extra means of making it easier to access and less so, say, less of a time gap between saying, okay, you see the output. You don't have to then download, but if you want to, you can.
Can we also download those like visualizations?
The containers themselves?
Also like, so like you were saying the output, and you can do it directly in the CBRAIN interface and we download those data, but can we also download like the visualizations?
Yeah, that's all within the HTML. In that case, for Cell Ranger, that's a visualization directly embedded within the HTML file. So by downloading that file, you're downloading what you see visually represented there. So it's not a representation of data that's only sitting in CBRAIN. So that's why when we say the tools, if the tool is crafting some form of output that is relevance for you, and it's displaying it through just a plain HTML file, yeah, absolutely, you will have that same visibility within it once you download it yourself. We try and keep I would say our fingers out of the tool as much as we can in terms of letting it do its thing. There's other things we do that try and maybe say extract information from the data in some cases, but those are separate things that we have on it, and those are more modality specific. And then there's other ones that other single cell ones. There's a pipeline of set of tools that we're about 90% of the way through integrating within CBRAIN, which something called scRNA box. So it's an scRNA-seq pipeline. We know the first 4 steps have been validated, but usually with this particular one, you don't run all 8 steps in linear sequential order and in an automated fashion. Usually it involves some form of inspection after each step just to do a sanity check of the outputs. Depends again on the tool, the nature of the data you're working with. Some of them you may want the pipeline to run straight through the end. Other ones you'll want to do a visual inspection. With CBRAIN, you can actually then just pick and choose which phase and which step you want to run and control the parameters. So for example, in the SA box input, this may not show too well, I apologize, really small, but you'll go through the exercise, you pick your file, that's what's shown on the bottom here, you pick your file, your FASTQ file of interest, you click launch. What you're going to do then is you're going to get your menu bar. You can then, depending on the type of data, select it from the menu of options, or just do a basic search by the name of the tool. Once you find it, so in this case it was scRNA-seq, or scRNA box, excuse me. Then the nice thing is, because of this boutique descriptor that I talked about before, it pre-populates the information. So anything that you would have normally seen as an option, you would have to figure out on the command line. You might be missing, put 2 dashes instead of a single dash. All this is taken care of through the web interface. So that way, things that are mandatory are very clearly marked. What the options are, if there are a variety of options to choose from, those are accessible from a drop-down menu, and you can then pick those accordingly. And all these steps and all these values that you take and set within the web UI version of the tool, those are all saved and basically in what are called task IDs, task information. So that way, if you need to rerun it again, or you want to look at it and say, well, what did I run this one with? All that task and parameter sets that you've signed are all kept within the environment. It's all kept within the logs, and you can know back and then rerun it if needed, or if you need to then say pointer to it to your colleagues, everything there information wise is stored. Same thing with the outputs, but we'll go through some of this in some of the exercises. So yeah, skip that one because you'll be doing some of that. So just to mention, as I said, everything from the web UI part, you can also do programmatically. So whether it's logging in to create a session, picking and working tools, submitting and managing tasks, and even working and handling your data, all that you can do from the command line, or say, for example, within a Jupyter notebook. And this is just an example of what we were doing in a recent tutorial that we did, where people were doing things with NiLearn. They were pulling the data in, able to visualize it all directly within the notebook, and they could push even things back into CBRAIN. Same thing, we could do that further. And we have, like I said, some examples already on our GitHub that show on how to go and create a session, doing all the workings with the data, selecting tools and so on. So those are all described on our GitHub pages. You mentioned earlier about question about connected resources. So this gives you, I would say, the breadth of systems that are connected to CBRAIN and where people can dispatch workloads if they are themselves permitted to run on those systems. So the Canadian-based ones, like I said, it's open to all people with CBRAIN accounts, but the other ones are more project specific. So we have collaborations and partnerships with other institutions that bring other resources in. So for specific compute needs or certain study needs, the access is more, I would say, controlled in terms of who can access those resources. So for example, many of the US-based systems that we see at Pittsburgh CBRAIN Center, we have other ones at the University of Minnesota. Those are used for specific collaborations where they're using CBRAIN to manage all their workloads for the particular study. Particularly in the case, if you might have heard of the HBCD project, Human Brain Development Project, that one is using CBRAIN to manage all their workloads. You can see obviously we're connected also into the ULICH system. We did that as proof of concept as part of HIBALL, and that's connected. And if you have a specific allocation in that system, it can be then configured to allow access for you and your group within CBRAIN as well. And the same thing we've done also proof of concepts. And even though all the systems here, the accesses are free, of course, the researcher to get them, depending on the needs as well, some organizations can also use it connected to our cloud, commercial clouds, but effectively works the same. We have over 1,700 users connecting over to the platform thus far. You can see this is at least the growth, because not everyone's using the system at the same time, right? We have on average about 400 active users in a given year. They might do something, go away for a bit, after things are published, have to come back, do some other work. So it varies as a function of time, and it's not just, of course, all Canadian users, it's worldwide. Touching upon just the accessible data sets, while it may be good that you want to upload data to the system that you yourself are bringing, we try and also make sure that the opportunity is there to save on the effort of having access to those data sets. So that's why we connect to different repositories, whether it's, like I mentioned, for the Canadian Open Neuroscience Platform. We also make it a common and easy place for people to work with more project-specific data, such as HBCD and ABCD, rather than each group downloading it a single time for themselves and having duplications of that data. We provide it in one common location, which then all projects can access, or all participants in the research can access. And that also applies to things like the Open Neuro data sets. Just before OHBM this year, it was turned on within the Open Neuro interface. So if you find a data set that you're interested in that's within Open Neuro, you go analyze this data set. You'll see CBRAIN amongst a variety of other different computing platforms now that actually can interface and see that data directly. And this is something that you don't have to ask us to particularly do the configuration for. It's all automated. So if you find a data set of interest, you go click, analyze in CBRAIN. It'll tell you if it's already been configured. And if it hasn't been, you just have to enter a couple bits of information, and then it'll automate the process of it making it visible within CBRAIN. So I encourage you, if anybody's working with Open Neuro data sets, to look at that. And as I mentioned, we also have one using the prototype in place for the eBrains connectivity. So anything that's from a HIBALL or BigBrain data set that's in eBrains can also be visible within CBRAIN as well. And that one should be out. We're just finishing one particular tweak and adjustment to the UI interface that'll be available. Anybody working with Open Neuro data sets currently? Any chance? Is your data set within eBrains? Neither. Okay. So there's not a group that's interested in it. Would you be bringing your own data sets into CBRAIN?
Into eBrains.
Into eBrains first. Okay. So if it's into eBrains, then you would then be able to, like I said, have it visible within CBRAIN. You wouldn't have to make a separate copy and say, well, I want to keep a copy in eBrains and I have to do the same thing in CBRAIN. No, you won't have to. Okay. You just have the one copy. That's it. Okay. And this is just to wrap up, say that the opening part, as I said, this is part of just a broader ecosystem of tools that have been built as part of an infrastructure within the Healthy Brains, Healthy Lives project, something called NeuroHub, which is think of it as more like a utility company, right? We're providing resources not only for capturing and managing the data, but also to do the processing and help people share it and make it visible to others so that they can then do their exploration of the data, but then at the same time, there's also connected pieces that will let you even publish the data.
And also, I see that you have some collaborations with the UK Biobank. Is it the digital biobank, or is it also the physical one?
No, they have the ones with the physical samples. So in this case, it's only the data. So we have, as part of, with the UK Biobank, what they do is they normally on a project by project basis issue essentially a data use agreement, and it would provide access to the data for that particular need.
Okay.
What since 2021 we've had is more what they would call an omnibus access, so access to all the data. That being said, starting a few years ago, and particularly they made a change of direction in July of this year, where they're removing, unfortunately, and I know there's a lot of members of the community not very happy with the decision and trying to voice some displeasure with it. The UK Bank also has an agreement with a company called DNA Nexus for their research analysis platform to centralize and make that the only entry point to get access to UK Biobank data. That was just new as of July, like on July 5th, they announced that, and that caught lots of people by surprise. So in light of that, still, there's obviously conversation, but this is specifically the UK Biobank instance for the McGill University collaborators for all the data that we are allowed to have access to, which is everything. The big asterisk now because of this change in the policy of the UK Biobank. If something like this will be able to continue after the end of our term is to be determined.
So it means you can't analyze it.
They would want people to go through the DNA Nexus portal entry where you would have to analyze it directly within their AWS based resource, and you would have to pay money to actually run the analysis resource. Whereas models like what we had in this case here, if you are, say, from Canada or from other institution that has high performance computing resources that were invested into by their national governments or other organizations, the effort was on the project to manage how that data was brought in, but there were investments made, but now it's being brought out to, there's lots of, I would say, security and privacy arguments that they're bringing forward as the rationale behind it. I should say, it's like I said, it's in a bit of a flux at the moment. But I know that, like I said, a large number of the community are displeased. But that's the UK Biobank example. Like I said, in our case, what we did go through is we took all the data, we even bitsified it ourselves, which is not something the format that you normally get in the UK Biobank. You get it in their format, but we bitsified it, we make it available, we pre-processed it so that none of our users had to do it. Yeah. So we did it once, then instead of 20 people having to redo the same compute, the same way, the same time, and multiplying all that cost, if you will. So this is I'll leave this one for you just to acknowledge the effort that CBRAIN's been around since 2009. It's not, a static development. It's been in progress and changing and evolving since the time. And one of the even named, common, I'd say statements from Alan as principal investigator of the project is that, the CBRAIN name was his choice. And he said, now maybe you would have second thoughts about calling it CBRAIN because of just I'd say the breadth of tools that are going outside what would normally be considered neuroimaging, for example. There's a lot more flexibility there. Are there any general questions before we go into the hands-on? And like I said, you'll get some flavor for the UI experience with the next basic exercises. Are there any general questions? I might have questions of you. Like, where is your data right now? You mentioned eBrains is where it's going to go.
Mm-hmm. So actually, due to the size, it won't be hosted on eBrain, it will be mainly reworked.
Okay. And otherwise, people have data stored where? Where do you keep your data?
Google storage.
Google storage, okay. How about so a departmental, some server that sits there, okay. Anybody else? Departmental servers? Okay. The nice thing, like I said, just to think about, even for the compute, even if you don't have compute, but then you have data storage, if it's a Linux-based system, you can make that visible to CBRAIN for your particular use. We actually have something that I can show you afterwards, basically what are called data providers. You could configure your storage to be a data provider within CBRAIN, so that if you have compute somewhere else but you don't want to move your data there, this can help make it easier to do your workflow. But I'm jumping ahead. I don't want to go through that too much at the moment. So we're going to go through some exercises. I just want to keep track of time, so we're okay at the moment still. So we're going to do some exercises just to get you familiar with working with CBRAIN, but also we're going to use it in the context of the BigBrain data. Okay. So at first, we're going to go through, see how far we can get. See if we can get through at least number 5 today. Number 6 is to save more, an optional one. We're going to go through 4 different things, really just getting set up and connected in. You're going to use a tool called BigBrain Workshop, which is really it's just an example. The tool itself can do a lot of things, but in this case, what we're going to use it for is just to show the workflow that you would go through of selecting data, processing it, and then seeing the output. And then you're going to watch and see how that status changes when you run the actual tests. And then you're going to look at some of the output, okay? So first thing, if you don't have an account already on CBRAIN, that's fine. Just go to the URL that's at the bottom there. I know I don't have it as a QR code. But right now, if you just, for the exercises go through, everyone will have their own account. So you just go there and just fill in your name and email. It's not going to get shared with anybody. What it will do is I'm going to assign each person basically one of those accounts to see which one you are, and then you'll get those accounts. So just for now, we'll let everyone take a minute and go through that URL and just sign up. And then maybe once you're done, we'll do an inventory just to make sure for those who are signing up gotten through the process. Okay. I'll leave that up there so people can type it in. Okay, everyone looks like they're good. Okay, let's do this then. So I'm just going to see it. Here's the name, so I'm going to zoom in here. It's not going to be, we'll edit these out of the course of the recording, right? So if it gets shared on the screen, this should be seen locally. It won't be shared with the outside people, which is good. So if you find your name here, and just keep track of the number here. So you're going to be, someone went from 2 to 8. Okay. That means, so does everybody remember their number? That means you're going to be HB0, whatever your number is. If you're already beyond, no one's beyond 10. So you're going to be HB0 and your number. That's your account name. Okay. And did I remove it? Of course. So you're going to go to this web address now, keeping in mind that you have your account name, HB0, whatever. Go to portal.neurohub.ca. Okay. And for the username, you're going to use that HB0 account number. And then the password is just going to be BigBrain and the exact same number. So if you're 03, it's going to be BigBrain 03. If you succeed in going through that and then logging in through the sign-in page, just maybe wave your hand at me once you've managed to go and sign. So thumbs up, hands up. Anybody else managed to get in and log in with their account? Because I know there was, I'd say 8 of you, but one of those 8 is me. Let's see it late. Number 9 now.
So I see the note about the BigBrain, so it's the thing that was what we did at home. That doesn't note about the
Exactly. We publish news things to make people aware. Of course, with these HB accounts that you have, you're not able to get access to the data, but it just was awareness for those people who are part of the UK Biobank. Exactly.
Yeah.
So anybody not logged in yet? Okay. So everybody's good. Okay, great. So everybody's inside. So then what you should be seeing is a portal view like this. This one, beyond what's on the screen here, is not exactly what you're going to see. You're going to see one that's a little bit more up to date. We usually publish a little bit, maybe more system status news if there's some specific information we need to convey to people, like a maintenance period or something like that, or as was mentioned already, UK Biobank outputs. You'll also see a different view sometimes if you run any tasks. So the real main thing is the menu. So this is the dashboard view that you're seeing right now. Right? Just gives you an idea of news, some of your task lists. There's other buttons that are going to describe what projects you have access to. Storage, which is more toward what I mentioned before. If you want to bring your own storage resource to connect into CBRAIN, that's the button you would use to connect it in. Okay. And then there's things like your account, if you wanted to change your password and things like that. Then we also have links to all our documentation. So those, the menu bar at the top, but what we're gonna go through is go through the exercise of seeing, one of the links that's on the left should talk about providing a link. You might have to scroll depending on things. We provide a link to all the data sets that are visible within the CBRAIN. Pardon me, I say NeuroHub, the platform, but it's essentially the same as CBRAIN for this, from that point of view. So if you go to, does everyone should be able to find a link? If you say click here, you should then get a list which shows two tabs. One that'll show you the tools. Excuse me, the tools, and the other one will be all the data sets. So if you ever want to just say do a quick check, you don't have to actually log in to see the tools. This is a bit small on the screen here. But you can easily then see what things are already configured from a tool description point of view, as well as from a data. And it also gives you an idea of what is generally accessible. Because sometimes we have data sets that are, that have different data use agreements, and therefore we have to, of course, make them accessible to only specific members of those projects. Okay. So if you can see the data sets tab, and you'll see one that's called, of course, there's multiple ones, which are more sub data sets within the BigBrain perspective. So you should be all seeing one called CONP-BigBrain. Okay. And if you go and then click on the more, you will see just a high level summary what it is there. But you can see BigBrain is one of the data sets there. So if you go back to the portal, you can also just do some discovery by searching through the search menu on the top right. Okay, so if you go to the top menu, on the top right, you'll be able to search there. But one of the first things we're going to do is you're not just going to say start doing some analysis. You say, well, I have a project, I have a specific activity in mind that I want to do with CBRAIN. So you're going to go to the projects button and you're going to create what's called a project. You can have multiple projects. You don't have to have just one. So if you click on projects, you should see a menu that presents you a visual view of what projects, which many times corresponds to a data set that you have access to. Okay. So if you go there, you'll see ones maybe not similar as this list. In my particular case, I have the UK Biobank there because I'm a member of the collaboration that's able to access the data. But if you add project, what we're going to do is add a project specific to the exercises we're going to do today. So if you go down to add project, click on that button, and then it's going to ask you for some information, just how you want to name that project. Okay. And then you give it a short description. There's usually a couple other variables that we ask people to check depending on their particular needs, but for this exercise, you don't have to do anything. So you just give it a name and a description, and then you click create afterwards. So let's go through the exercise. Let's create a project. You can call it whatever you want. This is your, if it's the BigBrain excursion that you're doing, can be something, it's only gonna be visible to you in this particular account, so you can call it whatever you'd like. And once you've created it, it'll pop at the top of the screen that it created that project successfully, and then you'll be able to see it there. Has everybody created a project? Okay, so if you create the project, that immediately brings you to that project view, and it'll tell you at a high level summary what's in that project. Yeah, of course, since you just created it, there's not going to be any files in it yet. There's not going to be any particular processings that you did with it yet, but you're going to see that there's one member, and that's going to be you. That's because you created the project, you're the member. There are options that let you invite other people to collaborate, so that way you can then each have access to the same sets of data. You can each have access even to the same processing tasks that you run on the data. So there's a lot of collaboration capabilities here. But what we're going to do with that now is actually go back to the dashboard, and we're going to say, okay, great. I've created a project that I intend to use for my exercises with the BigBrain data. Go back to the dashboard. So what we're going to do now is see just another way of just getting, let's say, browsing to the data. You're going to go to the top right search window, just type in, because we saw CONP-BigBrain is the main data set we want to work with. Just in the top right search box, just type that exact string in there in the search window, and you can hit return. So CONP-BigBrain in the search box, and then you'll get the search results. Will come back to you in that window. That'll show you everything that matches it. So it's a very, wide search. So you'll see pretty much all the results that you saw in the general data set list. But of course, these are the ones that match the search criteria that you ran. Okay. If everyone Is anyone not gotten that far? Okay, so if you, yep. Oops, I'll get rid of, I just wanted to see the zoom. Yeah, I'm filling the screen with the slide. Yeah. So we'll click on the CONP BigBrain. So I'll zoom in again each next time. So this shows you the information, the same high-level summary for that particular project, but much akin to what you saw when the project created. So you say there's 317 files. We're going to click on the files list. Okay, and then it'll present to you the structure of the entries within that data set. So here you can see out of those 317 files, it'll show you the first page of that listing. You say, well, this is kind of not very helpful. You're going to see a long list of 317 different files and different formats and so on. But at least it shows you some of the content. You can tell some of them are nifty files, some of them are MINC files, and then it also on the right-hand side shows you the sizes. So that way you can get some information about them. What we're going to do though, now we want to work with them. We're going to just, again, NeuroHub is the, I would say, the broader ecosystem portal. We're going to switch to CBRAIN. So in the top left, you're going to see this word letter CB, where you're going to go to CBRAIN. So if you go there, you should then get the CBRAIN specific view of that COP data set. So what you'll see is that you're in CBRAIN, you're still looking at the CONP-BigBrain project, you're still looking at the files, but now you're seeing it within the portal view. It looks very similar, but you should see the same thing. Does everybody see that? Okay. But now you're like, okay, well, great. I'm working with 13 pages of files. That's not very helpful, right? But what we're going to do is you're going to say, well, I want to work with a subset of data. Now, this requires, of course, you always need to have an understanding of what your data is, and what your data structure is like. In the case of the BigBrain files, in this case, what we want to do is run some analysis with one of the tools. But we're going to look at, though, just one particular format. So you can see across the menu list of options here in CBRAIN, it's not only the file name, but there's also file type, for example. So within the file type, if you click on the little magnifying glass, you'll actually then see the breakdown in terms of the numbers of files for each given file type. In this particular case, we're going to work at, say, just want to look at the MINC files. So if you click on the number beside where it says MINC files in that menu choice, you'll get a subset of that list.
And what's the main file?
It's just the one format, just a particular data format structure within that. You can see there's other ones which are nifty, MINC, depending on the particular choice of how the information is packed in the file. But what we're going to do is a further within CBRAIN, you have the ability to also do what are called filters. So using the selection of file type is one type of filter, okay? With the BigBrain data, you can also then do other searches within this, again, using the search box. For the one we want to work with in these exercises, we're going to look at the 8-bit grayscale images. We want to look at those. So within the search field, so you're going to do two things. You're going to select the 156 MINC file option to get just exclusively the MINC files. Then you're going to also type in full 8 underscore. So that means you're going to be look at the 8-bit grayscale images. Okay. And what you're going to end up seeing within your CBRAIN view is the reduced list of files that correspond to the filter that you just applied. You're going to see only the MINC files, and you're going to see only the ones that correspondingly have a name called full 8 underscore in them. Right? And then you can always see nicely, partway down from the top within the CBRAIN view, the list of filters that you're running. So you can see right at the top, the active filters are it's MINC file and full 8. Okay, so this way you know how you got there. You started with a larger set of files as part of your data set. You applied some filters to give you a selection, but at least then you can visually see what the selections are. Okay. So what we're going to do, so any questions about seeing and I would say selecting files at the moment based upon this example? Okay. So what we're going to do now is actually, you say, well, we've picked the file that I want to look at. I want to just visualize it within the browser first. So we're not going to analyze this yet, but we want to just take a look at it, right? And this is where, again, embedded visualization tools capable from the Brain browser that are part of CBRAIN will come into place. So what you're going to do, you don't have to put the checkbox there, but let's just say you pick any one of the Ming files that you want. But if you pick, say, in my case, I picked the full 8, the 200 micron example, or the data set, and this is where other people can better explain to me what went into creating what was called the optically balanced portion of it. It's much better described on the website for the data set. So if you just click on that particular file name, you're then going to get a view that presents a couple sets of information. One at the top, I'll zoom in a little bit just for people to take a look. Problem is it? I can't pan across, so I'll leave it. So you see the file name, you can tell us information about the size, when it was created, what project it's part of. But also down below, you should also see, it may take a bit to load because it's like a 70 some megabyte file, depending on the network here. It'll load directly within your browser, where you can see the volumetric view and can go through and traverse through the different slices from the different, planes of view. You can actually then visualize it directly within the browser. Okay, so you don't have to download it to another different tool. Okay, so you can view all that directly within CBRAIN. Okay. And you can also change, depending on the type of viewer that you have, we also have a surface viewer that's visible in there as well. Okay. So that's the nice thing, you can actually interact directly with the image that you're seeing without making any extra step to download. Okay, so now what you can do is you can just go back to the view. Now, if you back out, you may find that you've lost your filters. You can just reapply them again. Okay, so we want to get back to having only the Ming files in our view. And you want to be looking at the 8-bit grayscale images. So then you should be able to see this view that you still have that shows that subset of Ming files. Let me know if you get back to that page, or if you don't, then let me know. We can. Okay. So what we're going to do is, now that we visualized it, we want to run some analysis. So this is where you put the check box beside the file that you want to process. So what we're going to do is one of the exercises where we're just going to do analysis processing on one file. But then the process is almost identical if you want to process multiple files at the same time. But we're just going to go through the exercise of launching a tool on this singular file. So you select with the checkbox beside it, the full 8, the 200 micron file. Okay. And now once you've done that checkbox, you're going to actually then, what you're actually going to go through is click on launch on the top right. Okay. What that's going to do is pop up a menu options to you. You're going to see the select tool options list. You'll see it organized by type. You'll see it organized by different modalities. But what you can do directly though within the search box, because you know the name of the tool, like we're going to be running BigBrain Warp. So you just type in the search box BigBrain, it should auto-complete and show you then an option of what tools match the corresponding search criteria text that you put in. Okay. So if you type in BigBrain, you'll see BigBrain Warp, and then you'll see as part of a specific category. It may not look exactly as to what I have on my screen here because we've updated a lot of tags recently. You'll see which version of the software is installed. You'll see where that software is installed. So in this case, you're going to see it on probably just one system. But depending on the others, you'll see it visible on all the connected HP resources that you have access to. But in this case, you're going to probably just see it on one. So what you're going to do is you're going to say, okay, great, I've got BigBrain Warp. It's in the inventory CBRAIN is giving me. I'm going to click on launch the tool, launch BigBrain Warp. And it'll bring you to a menu where you're going to specify 2 different types of settings. The first one is going to be related to the CBRAIN orchestration settings, meaning how do you want to manage the actual task. The second one is going to be about the actual scientific tools parameters itself. Okay. So once you click on launch task, you should get this type of menu controls called task control. Does everybody see that in their information? So you're going to want to then make decisions about where you're storing your data and into what project you're going to store that data. Okay, because typically you're not going to be working probably working on multiple different projects. You've created different projects within CBRAIN depending on your activities. So in this case, I won't go into too many of the complexities about what we have option-wise in terms of storage types. But what you're going to do is pick main store. It's just kind of like the general purpose storage resource destination that we have within CBRAIN. So it's not the storage at your local lab, but it's just one that we provide to the community, some general amount of space. So if you go through and save your results, mark it to main store. Okay. And then for the next one though, you're going to want to select for the project, select the project name that you created at the beginning of the exercise. In my case, I just had one already configured that I created, but for your project, just select the one that you created earlier. Okay, and if you want, as part of, you can give it a description to help you, trace through or manage between your main projects. You can say, okay, this is the exercises we're doing as part of the tutorial, for example. Okay. So that's the task control part. That's the part that manages the workflow within the CBRAIN point of view. But next, you're going to get the set of parameters that the tool is expecting you to normally do. This you would normally, if you're running this in the command line, you're typing all of the different flag options. You're typing with all the different menu choices, which be written down in some documentation or in a help page or on the GitHub documentation for the tool. What it is here in CBRAIN is presented as a set of menu choices. Some of them are from drop-down menus. So for example, the input space. For this particular exercise, you're wanting to be choosing that the input space for the file that you've selected to be the ICBM space. So if you click the arrow, you'll see a variety of different options. But in this case, we want to pick ICBM, okay? Same thing with the output. Normally, if you're typing on the command line, you're expected to type BigBrain Sim. Here again, the arrow down, you can select BigBrain Sim from the menu of options. Those are the ones that are really key to the choice. Okay. It asks you to give a descriptor for the input. Basically, it's just something that will help you identify the workflow. So in this case, I just said it was part of my test exercise that I ran on my date. I put the -200 because it's, well, I'm running this in my 200 micron file. It could be any text that helps you keep track of your logical, I would say, structure of your data. And then because part of BigBrain Work includes running of FreeSurfer, normally you would have to go through and get your own FreeSurfer license, but for the purposes of the exercise, we've included one for these accounts here today. Okay, so you should be able to actually either see that field already pre-populated or with the arrow, see it and select the file FreeSurfer license.txt. So that's already given there for your tests today. Okay. So I've walked you through those, and there's one last one now, because you remember you did the checkbox in the prior page, right, where you looked at all the Ming files, you already put the checkbox. Normally what you would do, you'd have to type in what is the name of the input file, and you would write that long name, folate _ 200 micron. Within that box, it should already be pre-filled with the checkbox, the name of the file that you put the checkbox beside, okay? So that way you made the logical connection between what you clicked on and what the input is. So that way you're not making a typing mistake, potentially, right? It's all pre-populated for you. Okay. The other ones are more optional. So any of the parameters that are mandatory are marked with a red asterisk. Everything else is optional. But the nice thing is, as I said, you don't have to type in anything pretty much manually, anything other than to try and help you understand. So it provides that direct selection of the values that are needed. So then we're going to run start BigBrain Work. So now that's where it actually then does a submission of the task. The actual processing task will do that conversion from ICBM space to the BigBrain space for the input file that you selected. Okay. Then you should see when you've done that submission, you should provide a green bar at the top that says launching. Okay. Maybe people can just raise their hand when they've already submitted launch and they see the particular task running. So I see a couple. But you should see, particularly in your case, you will have only see one entry. In my case, I'm showing here how they'll show multiple tasks in my collection of tasks that I've submitted to CBRAIN for this project. At that point in time, what I'm showing here on the screen, you can see there's also completed tasks. Those are marked in green as completed. But the first thing you should see is that the current status of the task that you just submitted will be marked as new, meaning just fully brand new got submitted to the system. But within CBRAIN is that we do periodic checks of where the job is through its life cycle, okay? So what you're gonna see at various times, it's gonna change, not only from new, it'll be seeing things like preparing. It also then eventually, once it gets on the compute part, when it's actually doing the actual computation, you'll see what it says on CPU.
It actually doesn't appear in my face. I have found the message functioning, but now it doesn't appear.
Okay, what is your project? If you look next to CBRAIN here,
Yeah.
what is the name of your project? You'll have to, if you click on where, in my case, where it says all, because you submitted it as part of your project name that you created at the beginning, select that project. And then make sure you go to the tasks menu, and then you should be able to see it there.
Yeah.
Okay, good. Has everybody been able to submit a task, or it's in some form submitted and set it as green? Okay, so it's on CPU, and it may take a few minutes, but you'll see it go through that cycle. Like I said, you can actually then see. Now, of course, you don't want to be necessarily refreshing the screen, so all you have to do is refresh the page, and then you'll see the page cycle change. Probably take a few minutes. Because there's a few of you submitting the tasks to the same system that's been in computation. But eventually you'll then see it running from on CPU, changing from on CPU to finalizing and then to completed. Okay. And then you'll get another, once it does refresh and complete, you'll see then another message at the top in green saying, okay, that task that you submitted has completed successfully. And of course, sometimes though, you will get messages that say the task failed. The nice thing, even though it's a failed task, is that all of the logs that you'll see are recorded here. So even if the task is successful or failed to process completely properly, you can actually go through and see the log files. You can see how far it went through the task. You can see if it's an issue in terms of the task being assigned by CBRAIN to reach the system, for example, where you told it to run. Or if it's a problem, particularly in the scientific tool, sometimes it has a problematic input file and crashes. You can at least see all the logs, whether it's how CBRAIN orchestrated things, but you can also see all the logs from the scientific tool. All that's visible and stored within the system. Okay. Has anybody got it to a completed state yet? Got it completed? Okay, great. So what we're going to do is then now say, well, okay, great, I've gone through the cycle. I want to look at the output. Okay, so beside on that row where you see completed, you can pick your task. Under the task type, it's a link. So you can actually then just click on the actual task type for that one that you ran. And it'll bring you to a view of the output. Okay, but it'll give you some high level information at the top, indicating, okay, what is the task type that you ran, right? So that way, again, if you're running multiple tools, you're going to want to sometimes go back. I want to look at my BigBrain work test. I want to look at my track the flow files. I want to look at my FMR prep. You can see the explicitly different types. You can tell which version of the tool you ran, right? So this is going to be very important, particularly if new releases of the tool come out. You want to know, okay, which version are you dealing with? Because sometimes the version will make a big difference, right? You want to know how many times you ran it. It'll tell you how much of the output was produced and so on. So it gives you all kinds of information just to manage that output data. It'll provide details further underneath, like summary, like I said, of how long it ran and what the input parameters that you set for. Again, part of that important relationship between input data, the task, and the output, right? And then even if you click on the output directory, you can actually then go and do through things, and you see not only the log files like I'm showing here on the screen, you can see the very detailed logs, but if you click on the output directory, you can then browse to the content of the output itself. Okay. So if you click on the output directory, then what you can then see is the contents. And this is, for example, when I ran the output of the task for me, BigBrain Work on this 200 micron file, I can see directly the output that was produced. That's particularly in this case, the middle file. And then I can directly, because of the built-in viewer, all I have to do is click on that file itself, and it will then display directly within the browser. So again, you don't have to download it to do the viewing. You can see the relationship, you can see the logs, all that information is tracked for you within CBRAIN. Okay. And this is just an example of that particular one output. Of course, it's a very simple exercise in this case, but just to show you the end-to-end flow. Okay. And then one of the things that was mentioned before in terms of downloading files. So for example, if you wanted to download this output, you could do that. Or if you say, I only really cared about that particular file, you could do a checkbox beside any of those files, like the middle one, and you can say extract. It'll extract it as a singular file, and then you can download it from there. So there's lots of options and flexibility in terms of working with the output that's produced. Particularly with some tools, things like FMI Prep, for example, they have lots and lots of intermediate files that most times you don't want to keep, because otherwise it just becomes an organizational nightmare, and scientifically they're not of value. They were needed for the end of the running, but not for the output. Okay. Did anybody get an error in their running, or they were able to just run complete? Okay. Is it correct we have till around 2:30? Okay. I think the last thing that we'll do in the time that we have left is this exercise number 5, where we're going to say we're going to work with multiple files. Okay. So in this case, you say, great, you've gone through the exercises of picking one file, you've selected the tool that you would want to process it with, you've looked at the output. Now you want to do this on multiple files, right? Because most times you're going to be dealing with multiple files for your data set. Okay. So in this case, what you're going to do is jump a little bit ahead here, but what we're going to do is we're going to create another filter. So what I'd like you to do is go back to that data set for the COMP BigBrain, right? You should be able to find it in the list, or if you type it in the search box on the right-hand side. Okay, and what we're going to do is create what we call a filter, right? So again, you want to find, if you're not getting this immediate same view, you just go into the search box on the right-hand side, type CONP-BigBrain. Okay, and then you'll get, again, that list of 317 files. You're going to create 2 filters. Again, you're going to go through the file type. You want to just do, in this case, we're going to work with only the Ming files. There's this particular interest, so you then select Ming files. There's 156 normally. And then you're going to say, well, I want to work with just a block of these files particularly. And in this case, again, you'll have to know what your data is, but here I'm selecting just a subset, which is identified by the string called block 40-000. So you would type that in the search by name bar on the right-hand side. Okay, so you're applying 2 filters, make files, and the files that are going to have names, block 40-000. And then once you apply them, they'll show up in your active filters list at the top, like is in the red square here. And you should only see 9 files in the list. Are there any difficulties in doing the filter?
Yeah, I can go back to the project list, not the project list, but it's like the files. Yeah, the files.
Yeah, you can see, that's one way. You can either go to the search by name is the one piece, but you can do CONP-BigBrain search. There's other ways I can show you afterwards, if time permits, to how you can browse directly to the project.
Okay.
But you can do it in the search for anything, like CONP-BigBrain.
Because I have there's a project and I can switch to it.
Yeah, you click on the project, you can switch to it as well. Yeah.
Yeah.
We have that easy, what we call a fast task or a fast project switcher.
Okay.
Then you're gonna do the file type filter, so selecting only the Ming files from the magnifying glass. And then in the search by name box, type in the block 40-000. It just gets you a subset of the files. I'm just picking an arbitrary set that will give a reasonable number to display on the screen. But in this case, for any data set that you're working with, if there is more than many files, you don't then have to repeat. So are you able to get the 9 files?
Yep.
Okay, great. So what we can do then is say, great, I've gone through the exercise before of doing 1. You can just select the checkbox on top of the column corresponding to the column of checkboxes next to the file names, it'll select all of them, okay? Now you have two options here. One, you can actually then just go click and launch, but I'm just going to show you a different approach that creates what's called a CSV file or a CBRAIN CSV file. So what it means is that even though I put a checkbox beside all these files, I want them to be all treated the same way when I'm working with my analysis tool. So I'm going to create a CSV file that contains information about each of those files, and I'm going to pass the CSV as the input to the tool. So what you can do then, you go to the More option on the right-hand side. After you've selected those, if you click on More, you're going to see something in the bottom near the bump called Create a File List. Okay. So what, like I said, that does, it provides a way of creating, say, okay, I've identified what I want of interest. I want to put them all as a collection together of objects of interest that I want to process. And if you click on Create a File List, it'll then give you output showing you what the content of that file list is to say, okay, here's what you've done. Much of this, of course, is internal to CBRAIN, but it just gives you an idea of what it does. It actually then organizes all these files into a list that CBRAIN can then parse automatically, okay? So then with that list, you'll see that list appearing, or you should be seeing that list appearing inside your project directory that you created. So if you go to your project switcher, so next to the CBRAIN button, there's what in my case is called on BigBrain Research. You click here, switch to your project. You should see what's called that file list that have been created. Is it not showing up for you guys?
Now I can see my results from this.
Okay. Can you try switching to your, by default, everyone gets a project directory. So in your case, HB0, click on that one.
Oh, yeah, I can see it.
And that's where it is. Okay.
Should I move it to the other?
You don't have to, but
Is there a way to change where it default saves the file?
yes. So even in that case, if you click on it, so if you click on the file list, it'll open it up, and then under the info section at the top, it'll tell you what project it's part of. So in your case, if it does that, it might have just put it into your personal default project.
Yeah.
You can then switch it to be part of the project that you want it to be part of. So that way it keeps it organized. But for the purposes of what we need, it doesn't have to be in that organization. Okay. So now you've got the file list, and that's the only thing you have to then put the checkbox beside. You don't have to go through and, later on you say, okay, I want to run those exact same files through another version of the tool. I want to run those exact same files through a different tool. You can just you have that object stored showing what files you're interested in. You click the checkbox now. If you click beside that, and then you then select launch, and that's what you're going to want to launch. So if you select the file list and then go to launch, you will do the exact same exercise you did before. Okay. You're going to type in BigBrain in the search box, and you'll get the same menu appearance, right? It'll say BigBrain Work, version of August 2022. You're going to launch it on BigBrain Work on CBRAIN. You're going to have the same presentation of values you have to determine, meaning how do you want to launch the task? Meaning you're going to select where you save the result to. Again, you're going to pick main store. That's the main space of CBRAIN for storage. You can have it go to your project that you created at the beginning of the exercises. Okay, in your case, whatever the project name that you created. And then you're going to set the task parameters, just like you did the other one, right? But you're not doing this 10 times, or in this case, 9 times. You're only setting it once, and the parameters are being set to all the same for every file within that CBRAIN list. Okay. So in this case, you're going to again select the input space as ICDM space. Okay. You're going to also set the output space to be BigBrain Sim. And then you can give it again. The only thing that you have to maybe I say change or write something a little different is just the description that you want to type in for the description of the text, right? So in this case, I just marked it with a CVCSV saying, okay, this is what I ran on the CBRAIN CSV file. Everything else is already pre-filled out for you. So you don't have to worry about typing anything in the command line. And you can see that the input volume, instead of an actual main file, it's the actual list that you created with all the 9 main files in it, okay? And then all you have to do then after that is click the start CBRAIN work, BigBrain work, okay? Has anybody been reached that step where you then click, and then you should see the green launching of a task, but you'll notice something a little different this time, okay? You're gonna notice that you'll have, of course, your completed task that you ran before on the 1 input main file that you selected. But you're gonna see now the new, you're not just gonna see a singular new, you're gonna see the name of the task, there's a plus beside it, because this is a collection of tasks that you've launched. And you'll see on the current status, it'll say 9 times new, because there's 9 individual processing tasks that you've launched through 1 set of steps that you ran for, as if you were doing it for a single file, but you're running it for those 9 inputs, okay? And if you click on, the plus at any time, you can then get a deeper dive into those, but then you can see the same thing, even if it's running, you'll see the same cycle go through. You can see it going from like on CPU when it's actually running. Oops, back that way. And if you click the plus, it'll expand the information down. So then you can see the actual status information and details of each of the individual tasks that you've submitted, okay? You can see they may not, depending on where you're running it, depending on the type of tool, they will not be in lockstep with each other because they're all unique individual processing tasks, okay? And it'll tell you where they're run, so you get all that run information. Then you can follow them through and watch the status, again, refreshing the page. In a few minutes, you should actually then get it to a completed state, okay? But what we've done here really is, like I said, shows the, let's say, a simplified way of working with multiple files within CBRAIN. And it's as if you're only working with that 1 input. So you don't have, you're scaling up your workflow and your work activity, but you're not scaling up the amount of effort you put into the beginning part to say, okay, I'm gonna do this for each individual one. Or you're not needing to write a script on the compute system where you're trying to say, I'm gonna loop over this and do a S batch to submit it to the queuing system, or then I'm gonna run it once and then watch, make sure it finishes and then run the next one. You're benefiting from the automation that CBRAIN is bringing to offload that particular piece of managing the workflow to something else, right? So then you can just focus on the tool and what you need to do with the data afterwards, okay? So eventually, like I said, you'll see the 9 completed. Skipping ahead, and then you can even look back in your project, you'll see all the different output directories that get created by each one. Then you can do your post-analysis work. You can look at the output of any particular one. You can see the log files if any of them were to fail. You can double check and see, okay, even if there was, say, a failure because maybe you misset one of the parameters, you can then correct the parameter, relaunch. So all that diagnostic information is kept there for you. This is particularly useful if if you're working on it for a bit, you're dealing with another particular activity, and you come back to it and you say, okay, where was I? And you can see directly within CBRAIN, you know exactly the relationship again between the data, tool, and the output. So then you can see where you left off, what information was there. So it's saves a lot of information that you would normally have to take care of on your own. Okay. I think that's then I'd say the most logical place where we'll stop. Downloading is straightforward. You can either download through the browser, but then there's other, I'd say, different ways I'll include as part of the slide deck that will go on to the agenda page afterwards. The details if you want to do larger scale data upload and download. But this, I hope, should give you a feel for, at a high level, what CBRAIN can do across the range of data processing types and data types that it handles. But at the same time, gives you a flavor for the exercises going through and working with files, seeing the view, navigating through the system a little bit. There's a lot more things we could cover, like things like the notebooks, like I said, the APIs. I have links that I've given in here as well. But certainly if there's things that you'd like to see as extensions or if you're not sure of, I'm here for the full workshop. I'm happy to sit down and go through some of these with you. If there's any questions, or if you want to see how it possibly might apply to data you're already working with, right? You can just find me, and like I said, we can try some examples. The key thing is that as long as the tool can be put into a container format, then that makes it very much eligible then for including into CBRAIN as a tool. So if there's things that you see in the tool list inventory aren't there, let me know, and then we can look into including that in our inventory as well.
Can you tell us like, how you would open data set?
Sure. Yeah. I'm just going to switch out of this one because this focuses on the tool piece. So then I'll share my screen for another part. What's that? Yeah, I just share this with people outside so I can see it as well. Okay. So this is my view with one of these same HB accounts, okay? So what I can do, right? So in this case, and I didn't create yet a project, but I'm gonna say, I'm just gonna use my default project to upload the destination to. So there's a couple of ways of doing it. One is I just go into my project that I created. Of course, I got a lot of HB01. Oh, this is from when I ran it before. So say I want to upload a file that's local to my laptop right now. Okay. You'll see under the files menu. Where is it here? We have our upload button. Okay. It brings a little pop-up. Then you can decide. Okay, a couple things first we should do before selecting the local file. So much akin to what you saw in the task launching, you decide where you want to store it, meaning what type of storage. Everyone gets access to small amounts, I say small, just under 1 terabyte at the moment of space on main store. So that's the storage hardware. You decide what project you want to put it in. In my case, I only had the one project for this particular account. But if you had multiple ones, it would be appearing in the menu of choices you could bring it to. So all you do is you pick the storage location and the project name. And then you're going to browse to the particular file. Okay, and I have some files here locally on my system that may not be very large here. So in my case, I have someone, let's see here, I have files to upload just for this exact purpose. Say I have other files, I'm going to upload this one here because this is, again, some CBRAIN or BigBrain files. I have this, the 400 micron data. This is just a file. It could be anything that doesn't really matter. I click open, it selected the file. I click upload, and then it does it automatically through the browser. And the browser one will support up to around 600 megabytes. Now, it may not seem like a lot. It's just intended to, for best performance, we keep the size of what people can do for the browser for uploads to be small, okay? Now you say, okay, great, that's only one file. I've shown you. How do you do many files, like an entire data set, for example? So that's where our SFTP interface. So if you used SFTP before, so the Secure File Transfer Protocol, all we have to do, we have endpoint information about how to connect things into CBRAIN, and this is in a slide I didn't show. So anything that you use to manage your files from a transfer point of view. So in my case, I'm just using any open tool you can use. You can use a command line if you want. If you're comfortable with that. There's also one's called CyberDuck that I like to use. And within that, all you would have to do is tell it where within CBRAIN to connect. We have some specific host name number and location, okay? So let me just bring this up here. It's hidden behind here. So it may not be very large on the audience here. So already I have within my CyberDuck configuration, the SFTP input location for CBRAIN. Now it's my own personal account, so that I'll switch to that. That's fine. So all I have to do, yes, that one. I'll share my screen then. Bear with me as I switch. Share the full screen. There, that should be better. So what I'm going to do is I'm just going to say, find my data set of interest that I'm going to work with. In my case, I have in my files upload. So say I have, let's see here. Let's find something that's of interest to. This is one that was more, I was showing as an example before from the cardiac imaging. It doesn't matter really what it is, but it's a directory of files. Right, so if I go in here, you'll see it's a bunch of other directories, and I think in this case, it might have been a bunch of DICOMs. It doesn't really matter, but what I'm gonna do is say, well, this is the directory containing all the files I upload. All I have to do then, in my case, for CyberDuck, is I drag. Oops, let me drag it. Oh, it's gonna let me drag it. Then you can see, at least in, again, this is where we don't force people to use the command line to do the uploads. If you have a popular tool that you're more comfortable with, I know on Windows, there's one called, like, I think, FileZilla, something like that. So you can use that. You can see all of these files, and I'll have to interrupt it here because it's gonna take a long time over this network. So it's in transit now. So this is uploading to an SFTP area on CBRAIN. And then once it's finished, all you have to do is then move it into your project, because it's right now essentially being put into a parking lot, like a temporary space that's uploading to. Once it's finished, all you do is say, okay, I want to reassign this to my project that I uploaded. Then all those files that you uploaded will all be visible within CBRAIN for you to then run an analysis tool. So this is the preferred way of uploading data in bulk. The web interface, great for, a couple files, few files like that you can do one at a time. But for large bulk data, this is the way you submit it to the SFTP transfer, let it go and let it do its thing, and then you
You have
just.
some sort of user account in that SFTP, so.
Correct, yes. Yeah, so in this case, I just use one of my existing accounts, but in which case, if you want to use it with the HB one, it would have to be the credential for the HB account that you have. So it has to match, of course, the same account. It's not a generic one that everybody has access to. But by creating an account on CBRAIN, you get access to the same interface, and you can upload the file. Okay, so that's one way of doing, like I said, the bulk versus just individual files. Okay, like I said, we'll wrap it there because I want to keep everybody on time for the break in the next session. But like I said, I'm here the whole time. If there's any questions, like I said, you want to try some examples beyond what you just went through in the basic exercises, you want to maybe see about your data, particular tools that are maybe are not in the inventory, or if they are in inventory, how to run them. Either I would be able to help with those, or I could draw other people in who aren't from the team that aren't able to make it here. So then we'll be able to help with those. So I'm happy to hear more about what you're trying to do and see where CBRAIN can be of help to you. So thanks for your attention and thanks for your time. Stop this upload.
Okay, we have copy right now again, and then at 4 to 3, we will be back in the 2 rooms. There will be Amy and today about the BigBrain data set in the big room downstairs. And in this room, Timo and Sebastian will talk about Libra Python and the Python interface to the Libra SQL.
Hi, I have a question about data from the ADME database.
Okay.
So is that also in CBRAINs, or is that something like, for example, my lab, we use a lot of data from ADME. So would I have to download that first and put it in CBRAIN?
I can't remember what's the use agreement specific to the admin. Is it fully open, meaning people can just download it, or?
Yeah.
Without registering or any control access to it?
So I think you have to have like a registered account for it. I'm not sure how the registration process goes, so that's the PI.
I'll take a look. Yeah, I can't remember. Or it's just one that has to be downloaded normally through the NDA, in the US. I can't remember, or is that something so the NIH data archive also.
Yeah.
What data sets? I don't know if Ashley's in that one. I think it's separate.
I think it's separate, yeah. But yeah,
So what we do sometimes is, in the case, one of the ones that we do particularly for projects is that, I'll take an example, sounds similar to this. Human Connectome projects data. We have multiple researchers saying, okay, I'm interested in this data. And rather than them individually downloading it and then putting somewhere, we put it in a common place that's accessible to them. So we do it once and then multiple people get added to be able to access the data. particularly in that case for things like the Human Connectome project, they have to show us proof that they've been given permission
Yeah.
to look at it, and I think it might be the same for the item, at least to show that you're been authorized to do it. So we have the settings such that it permits us to have permissions controls on it. So if that was something that, you know, multiple groups or multiple researchers within one group say would want to access, we can make it visible either by on our system, but we also could put it such that, okay, if you've downloaded it to your storage, we can make that visible to CBRAIN such that you and your team can run tools on it from that. Because depending on the volume, we may not be enough capacity to host it in our office space, but if it's already in your space, we could just make it visible in CBRAIN. You understand? So as long as it's on a living space system that we can reach, we can then make that data visible to CBRAIN.
Perfect. Yeah, that sounds good.
Yeah,
Thank you.
yeah, no problem. So yeah, reach out and then we can maybe come
Yeah.
through and look at some examples and, you know, just see this, verify the structure and things like that. That way, it would then, like I said, be visible to you, and then you can work with the tools that are there and still point to the data you've already downloaded.
Yeah. Thank you so much.
You're welcome.